Friendly reminder, if you've heard anything interesting, you can tweet using the hashtag #KIMissionPossible, tag the Koch Institute. Sorry. So next up we have Project Asclepius, and they're coming to us from the Jackson Anderson Labs.
Come on up, guys. Megan, thank you. Judges, thank you. It's really a star-studded panel of judges, and I'm not just saying that to butter you up. It really is a star-studded panel. I'm here representing Dan Anderson and the rest of the members of Project Asclepius. And we have a bold idea. You've heard some very cool ideas already, new imaging techniques, the ability to target lymph nodes for invasive cancer detection, sequencing RNA from Bob Langer's poop. Those are bold ideas. We have a bolder idea, which is to develop technologies to prevent some or all cancers. So many years ago, a wise man, Benjamin Franklin, said, "An ounce of prevention is worth a pound of cure." And there's real truth in that statement. And to remind our judges of that truth, I've asked my colleagues on stage with me, David Canner, who's a graduate student in my lab, and Omar Khan, who's a postdoc in Dan Anderson's lab, to hand out some Benjamins to the judges in hopes that they'll remember. So there's probably no time and no disease for which that comment by Benjamin Franklin was more appropriate. The fact of the matter is that this year, 14 million people in the world will be diagnosed with cancer, and by current statistics, about 8 million of them will die from the disease. And by 2030, those numbers will double unless drastic action occurs, either at the treatment step, and we hope, obviously, for better treatments, or in the way of prevention. And that's what our goal is. Now, prevention in cancer is not a new idea. There have been efforts to prevent cancers for a long, long time. Some of these efforts are maddeningly difficult to implement, like making people stop smoking. Others are too narrow, like prophylactic vaccines for virally induced cancers, like HPV-induced cervical cancer. Some of them are simply barbaric, like the choice that a woman who's a BRCA1 or 2 carrier has to make to have a double mastectomy or a double oophorectomy to avoid developing mammary cancer, breast cancer, or ovarian cancer. We would like to create new possibilities to improve the likelihood that prevention can happen for not one, but perhaps all cancers, to give that woman a better choice and to change the curve for the incidence of cancer throughout the world. And that requires new technologies, which my colleagues will introduce.
Our concept is to create a protective vaccine, and in which we train the immune system to fight cancer as it would an infectious disease. To do that, we take advantage of 3 important components. The first is nanotechnology that allows us to rapidly create very potent vaccines for any disease. The second, we'll use genomic tools to actually sequence these cancers and find good vaccination targets. And finally, we use the world's most accurate lung tumor model to test and validate these vaccines. Project Asclepius exists at the interface of these 3 components, and that is what makes us unique and what allows us to make the seemingly impossible possible. When you're infected, whether it be from viruses or parasites, your immune system fights back. And when it does that, it creates cytotoxic T cells, which are great for fighting off active infections and killing them, and antibodies, which give you memory and prevents reinfection in the future. Vaccination is a powerful healthcare tool because it can shortcut this entire process and get you your T cells and your antibodies without ever having to be infected. So towards this end, we have created the world's best and most effective replicon nanoparticle vaccine platform ever. And this is how it works. We start off by identifying a disease, any disease. Then we find a unique protein on that disease. This is called an antigen. We'll use the antigen to train the immune system such that whenever it sees the antigen, it will know it's connected to a disease and destroy that disease. Once we identify the antigen, we sequence it to get the corresponding mRNA. We combine that with our replicon template to form our payload. And this is an instruction set, and it has two parts. The first part are instructions to create an enzyme for self-replication. The second are instructions to create the antigens, and that's what we're using to train the immune system with. We take that replicon and we combine it with an excipient and the novel part of this platform, which is a custom-designed delivery molecule. When these components are combined together through mixing, they self-assemble to rapidly produce our nanoparticle vaccines. We take these beautiful vaccines, and with a single intramuscular injection, they get inside the muscle cell. Once the nanoparticles are inside the muscle cell, they release the instruction set, the RNA replicon. This is then translated by the ribosomes, which are inside your cells. The ribosomes follow the two instructions, the first being to create the enzyme for self-replication, and the second to make the antigen. The enzyme makes more copies of the replicon, which in turn are translated by additional ribosomes in your cells. And this way, we achieve self-amplification and can achieve protection with one dose. In parallel, the ribosomes are also making the antigens, so we're producing a copious amount of that. These antigens are released from the muscle cells, they stimulate the immune system, and then we get our T cells and our antibodies. So this platform works. And you can see from these survival curves here that we are able to rapidly create nanoparticle vaccines against a broad variety of different pathogens. Deadly pathogens including Ebola virus, Toxoplasma gondii, which is a microorganism parasite that's a relative of malaria, and H1N1 influenza. When we vaccinate these animals and expose them to lethal levels of these diseases, we actually achieve 100% protection. And that happens with just a single dose. So now we have the opportunity to further broaden the utility of this platform and apply it to a new application, in this case, making a prophylactic cancer vaccine.
So the question really is, why would you want a prophylactic cancer vaccine? Well, the main answer to that is cancer is still a really big problem. As Tyler mentioned previously, about 8 million people will die from cancer this year globally. It'll cost in excess of $1 trillion in terms of overall economic burden. And in the next 15 to 20 years, it's only going to get worse. The incidence of cancer is expected to double. Furthermore, and is very relevant to this conference, or this event, most cancers are detected very, very late, limiting treatment options. And because of these big issues, the JAX lab and other labs throughout the past 20 years have been developing really sophisticated mouse models to basically understand how tumors develop and form. These mouse models, like the so-called KP model, which we use in the JAX lab, mimic the human disease incredibly well. So then you can ask the question, armed with these very powerful mouse models, is a prophylactic vaccine against cancer even feasible? Is it even worth trying? And quite frankly, Dogma, and when I started this project, would say no. It's absolutely not feasible. That's because of this thing called thymic tolerance. Basically, as the immune system matures, thymic tolerance eliminates those cells that would otherwise attack your own body. This basically prevents you from developing autoimmunity. Unfortunately, cancer is a disease of self, as was previously mentioned. Thanks for the alley-oop. And basically, your immune system has been trained to not recognize these cells, which would basically prevent a prophylactic vaccine from ever working, since tumor is a disease of self. Fortunately, over the last 15 years or so, thanks to a lot of the sequencing efforts, we've discovered that tumors actually are able to express unique genes that are typically not expressed at all in the adult body. One class of these genes, the so-called oncofetal proteins, are only expressed in, as the name lends credence to, the fetus. Turns out these genes are also dramatically upregulated in certain kinds of cancer. And this was exciting in and of its own. Eventually, though, especially very, very recently, groups have identified immune cells that can actually recognize cells that express these oncofetal proteins. And this was incredibly exciting. It basically means that since these oncofetal proteins are expressed in a broad variety of cancers, and the immune system is able to recognize it, it lends credence to the idea you could create a vaccine that would not work for a specific kind of cancer, but potentially all cancers and in all patients. And this excitement led to a lot of trials to be immediately undertaken. Most of these trials were done in a therapeutic vaccine scenario where a patient already has a tumor, and then you vaccinate to try to elicit a more robust immune response against that tumor. And unfortunately, most of these vaccination studies have not worked. And there's two major reasons. First, the tumors are able to create an immunosuppressive environment, which basically prevents the robust immune response you're trying to create from actually taking place. And second, most patients that actually get cancer are elderly, and it's very well established that elderly patients, elderly people in general, have a weakened immune system. So you're trying to create this robust immune response in a patient that already has a weakened immune system. It just doesn't work very well. But this raises an interesting question. Because of all the studies we've done on tumor progression in mouse models and in humans, we know what tumors are doing. We know how they develop. We know what genes they're going to express. And if we know that tumors undoubtedly are going to be expressing these oncofetal proteins, why not treat cancer the same way you treat an infectious disease? Why not prophylactically vaccinate a patient, basically arming the immune system with the ability to fight cancer before the cancer even appears? Prophylactic vaccination has a number of benefits compared to therapeutic vaccination. First, you're establishing this robust immune response and subsequent memory in a patient that does not have a tumor, does not have an immunosuppressive environment, and thus able to elicit a more robust response. And second, you can prophylactically vaccinate at any age. You don't have to wait for the person to become older and develop cancer. You can vaccinate at a time when their immune system is fully functional and develop a more robust response. So the way this would work is you take an oncofetal protein as your antigen, you'd load it into Omar's amazing vaccination system. This would eventually develop immune cells. An immune response would become memory cells. They'll go throughout the body, basically in search of putative and nascent tumors. As soon as a tumor begins to form, the immune cells will home to these tumors and destroy them before the tumors can grow and cause any morbidity. We wanted a proof of concept that this could actually work. It's kind of a high-end idea. So we wanted to validate this. And we decided to use the oncofetal protein HMGA2 as our antigen in this case. HMGA2 was recently discovered by the JAX lab to be incredibly highly upregulated in tumors and not expressed anywhere else in the body. So we incorporated HMGA2 into the replicon system Omar described, vaccinated mice, and subsequently induced tumors via orthotopic transplant of tumor cells into the mouse lungs. And really excitingly and somewhat surprisingly, it's worked incredibly well. Unvaccinated mice universally developed tumors ranging from small to large, whereas vaccinated mice didn't. The majority of mice developed no tumors at all, and those tumors that did form remained incredibly small and had effectively no effect on morbidity of the mouse. We are now repeating these results in the more robust KP model, and we're excited to see if and hope it does repeat, and we believe it will. Furthermore, HMGA2 was just a single antigen we selected originally. We're developing a pipeline to subsequently identify new antigens via transcriptional profiling of tumors from our KP model and bioinformatic analysis of the wealth of data that exists about normal tissues in the human body to try to identify genes that are uniquely expressed in tumors and not anywhere else in the adult body, with the ultimate goal of creating a multifocal, multi-antigen vaccine that tumor cells can't easily get around. So this kind of gets back to the original idea. To be perfectly honest, this graph kind of scares the hell out of us. Cancer's only going to get worse. And quite frankly, the goal of Project Asclepius is to reverse this trend. And really, in order for this to happen, we need to change the dialogue about cancer. We need to shift it away from figuring out new ways to treat cancer and shift it toward identifying new ways to prevent cancer from occurring in the first place. And this is what Project Asclepius is about. Using a powerful, proven nanotechnology vaccine system, using advanced genomic tools to identify new antigens, and using, as Omar described, the world's best lung cancer model to basically test these vaccines and see if they can work before progressing potentially into the clinic. And with that, I'd like to thank our other third team member, Just Dave Chahal, and all the people that supported this project, including the KI, for this great event.
So as I read through your proposal and as I listened to this, I had two thoughts in my head. The first is from a great man, which is, who am I to judge? And the second is from not a great man, I'm not worthy. That said, let me try three questions before I let better minds loose on you. The first is there's 14 current immunogenic lung vaccines on trial in humans between phase 1 and phase 3, 12 of which are recruiting. Is there something that you've learned from these 12 trials that's changing or informing what you're doing?
Yeah, so, and it really guides why we're aiming at the prophylactic setting. All the vaccines that you've been discussing are done in a therapeutic setting, once they already have tumors. And what we found almost universally from these therapeutic vaccines, in fact, only one vaccine to date has actually been approved, and honestly, nobody really uses that drug anymore, is that therapeutic vaccines really struggle to work. And it's because of two of the things I mentioned. Basically, you have this immunosuppressive environment that basically prevents eliciting a strong enough response. And basically, your immune system, by the time you're getting cancer at these older ages, is so weak, it's hard to elicit a new response. Vaccines elicit a new response more than anything else. And you just don't have the capacity to develop a new response once you're...
So none of these vaccines would induce an immune response?
They're able to induce an immune response, but it's not strong enough to overcome a fully formed tumor. In our context, we train the immune system to recognize the tumors when they're at the potentially single cell stage or very early adenomas and haven't developed this immunosuppressive environment.
If I can add, we actually have data from that same mouse model that if you take established tumors and try to vaccinate them, once the tumor exists, it's ineffective. But David showed you data that if you prophylactically vaccinate with this one antigen, you can prevent the disease. So that is consistent with what he just told you based on the clinical experience as well.
So the second thing I was curious about is vaccine trials are notoriously long and kind of expensive.
Yeah.
Would it be longer and more expensive to run a vaccine trial on a prophylactic, and would it be harder to recruit people than people who are already sick?
Well, there's the opportunity to actually do it in at-risk populations, like Tyler mentioned at the beginning, BRCA mutations, people who will get cancer. Then if we can prophylactically vaccinate them at a later age, then we have the opportunity to run a better clinical trial and actually get that out. So think about vaccinating at-risk populations rather than everybody and starting very young.
In addition to that, I'll just add, with patients that have Lynch syndrome, so mismatch repair deficiencies, either genetic or acquired, they undoubtedly will develop colorectal tumors. It's a given, and these people are screened very, very carefully, often getting colonoscopies in excess of once every 3 months. So if you were to do a prophylactic vaccination in this population, you'd very, very quickly be able to tell if you're having some sort of effect, because these patients, in a matter of just a couple years, often sooner, are developing these tumors or these early lesions. And you'd be able to see if you're, say, postponing these patients from getting these early adenomas or potentially more advanced tumors. So the clinical trial originally would be done in these at-risk populations because you can shorten the clinical efficacy. Also, there's a greater window for potential other issues because these patients undoubtedly are going to be getting cancer, and so it's kind of a lower risk.
And to put a number to it, there's probably somewhere between 600,000 and 800,000 BRCA1 and 2 carriers in this country alone, and probably half that number of Lynch syndrome patients. So it's not a small number of individuals who would actually be knocking down the door to sign up for such a trial.
And last question, are you talking to the people who've been doing the International AIDS Vaccine Initiative, where there's also been just a huge amount of research on vaccinations, and it almost seems like you need a rolling admissions type of trial that allows you to modify the vaccine and get a rapid approval for the modified vaccine as you learn more about mutations and changes. How do you learn anything from the AIDS effort, and how do you think about a rolling regulatory approval process?
So in conversations with the FDA, one of the things about this particular platform is that it uses mRNA. And right now, in terms of their legislation, they've only really caught up to DNA-based vaccines, which have their own challenges because it can integrate in the genome and cause cancer. And with the mRNA, they're just catching up. So they're actually more excited to work with us one-on-one and as we develop the assay and go through this. So it's actually quite uncharted territory. There are some other people out there, companies such as CureVac, they're making normal mRNA vaccines. Their vaccines take several doses to work as opposed to ours, which is just single dose. And, as one of my mentors ended up telling me is that companies like that are great because they take a few arrows in the back, and then you can benefit from that attrition. So that's great because now this is getting out there. People are understanding that vaccines have changed. 80 years ago is when we started to make vaccines with fertilized eggs. That process takes about 6 months. And the FDA is recognizing now that waiting that long to produce a vaccine, and also with technology changing, such as with nanotechnology, it's counterproductive to wait that long. So there are new mechanisms that are happening. And this is an ongoing conversation. So it's in flux, but we're very hopeful that things will continue to move quickly.
I'm sorry. Is that all right? I have to wrap up the Q&A. Thank you guys so much.
Because I mean, this is crazy enough that it's actually very exciting. Oh, so my question is, and the reason, just because it's such a like dorky question, but this whole thing rests on oncofetal re-expression. So how do you think about impact? When are you going to test for reproductive toxicity?
So that's a very important point, and it is an issue that extends even beyond just reproductive things because the issue of autoimmunity, and we are.
It's a different question, but this doesn't.
Yeah. So specifically for reproductive health, I think it is something that needs to be taken into account. It's conceivably possible that these vaccines wouldn't be compatible with a woman eventually basically giving birth to a child.
But these are testable ideas in preclinical models. So for sure, it has to be tested. It's an extremely important question. If there were a problem, it doesn't kill the idea because of what David just said. You could treat women who have chosen not to or of an age where they will not have children, or treat men. But we can test the idea in preclinical animal models.
And David didn't mention, but he's done a lot of work looking at alternatively spliced proteins as well. So there's always opportunity and a contingency to get around that.
I think it's also worth mentioning that if a mother is giving birth to a son, the son has an entire chromosome worth of genes that the immune system theoretically could be attacking that doesn't. There are mechanisms in place that protect the fetus from an immune response, the placenta along with regulatory T cells, et cetera, that you could theoretically envision a scenario where the fetus might actually remain protected, maybe with the help of immunosuppressants, where if this ended up being an issue, the mother could be fine. Something just needs to be tested.
Sorry, I just had to ask.